Thank you very much, and I'm Fred Apple. For full disclosure, I am on the board of HyTest, so just to make that disclosure. Maria asked me to, and HyTest asked me to give a talk about where the state of the art of the 25 years in celebration of their company. I did take a little different approach. I'm going to spend time telling the history of cardiac biomarkers and how HyTest came into the field and where they are today on the cardiac troponin side. I've shown this, this is a really nice cartoon I saw in Minneapolis in the healthcare system, and clearly where we have come with troponin assays based on some of the reagents and antibodies used by HyTest in developing high sensitivity assays is we want to be the heart attack comma. We want to be alive when we use these markers in detection of injury. And I have to show this up. These are my colleague Jordi Ordonez from Spain, and this is a picture of me from 1982. So we've come a long way. Jordi has studied natriuretic peptides, which we'll hear about later. So the world has changed from whatever this was, all right? So when I started in this field, when I started studying cardiac markers in 1982, I drew some blood. This is an old SMA 1260. And what I noticed right away, I had an increased total CK and I had an increased SGOT. And these used to be the markers of detection of heart attack. And I was fortunate to meet over the course of my training, some of you may know this is Art Carmen. He described the first marker for the detection of heart attack using AST SGOT. And this is Sidney Rosalki. He was the one that first described the use of total creatine kinase for the use of detection of MI. And I corralled them at an AACC meeting way back to get the picture with both of them. So where that came up, we noticed that use of creatine kinase had a lot of non-specificity. Everyone has an increase of CK, just like my initial SMA 60. What does it mean? So then we moved off to the era of isoenzymes. And this is actually a picture of, right here, of my isoenzymes. And you can see here my CKMB, which became the new tool in the early 80s, was at 9%. So what does that mean? The rule of thumb for those of you who've been around the field for a while, anything greater than 5% is a heart attack. Well, I was admitted as a fellow to the ICU because of my high CK and my increased CKMB. All right, and I'll get to that. And as you can see here, as a trick, I put a little piece of dirt on the electrophoretic gel and I got this little heart to show up, just like it would in CKBB. It turned out that it was from skeletal muscle. So we learned that it was a very non-specific marker in injury to skeletal muscle that released CKMB. But we moved from an immunoassay type on 2 sandwich antibodies to the world of Jack Ladenson, where he developed the first anti-CK monoclonal antibody against CKMB. This was his fellow Ham Vaeda, who worked with Jack to develop that monoclonal antibody. The field shifted quickly, and if you look back, we're talking this was in the 80s. And in the 90s, in 1995, if anyone was at the meeting in New Orleans, billboards all the way from the airport to the convention center first showed the release of the Boehringer Ingelheim, now Roche anti-cardiac troponin T assay developed by Hugo Cadis. And also Jack Ladenson developed probably the second troponin I assay in his lab with Geza Bodar developing the assay. So again, this is now in the mid-1960s. It's about this time, 25 years ago, where HyTest came into being. And the role of cardiac troponin, in this case a study we did early in 2004, the power of troponin shows on this 1,700 patients, 8.8% of the CKMB back then was a false positive. When you move over to, you see here just troponin, this is the early assays, troponin positive, CKMB negative. You can see here now we're getting a little more sensitive, and it drove clinicians crazy. So where are we today? I don't know if anyone came to my workshop this morning for the IFCC. People are nervous about high sensitivity assays because there's another shift. We went from MB to troponin, now troponin to high sensitivity. It's going to get better with that. You shouldn't be scared of high sensitivity with the antibodies being used because they're going to avoid the noise like we had with CKMB. And the reason this is so powerful, this is one slide from a study. I'm going to show some work from our lab. 1999, this is an ROC curve. It was pretty obvious, cardiac troponin, in this case cardiac troponin I. This was the old DADE assay, the DADE Stratus. Very powerful tool to show an improved diagnostics. So now everyone started gathering key opinion leaders, experts around the world. The IFCC, we had a task force. Mario Pantagini chaired it, and we gathered in, you can see the chemists gather in a cold winter day in Germany. And this doesn't show up here, but then the cardiologists got involved, and they published their first universal definition of myocardial infarction in 1999. The cardiologists, and I was fortunate to be invited as part of the biochemical working group, they meet in Nice, France in July, so they're much smarter than the chemists. But fast forward my relationship with HyTest. I met Alex Kutruka, who's the head of research in the Moscow research branch, and I met Maria Saravina. They were part of our IFCC group when we met, and we did have a little conference. We met, and you can see here's Maria and here's Alex. And we took a gondola up to some mountain that's in the southern Germany to have a beer, to talk troponin though, all right? And then currently, as I said, I chair this this committee, the Committee of Cardiac Biomarkers, and we've been trying to do educational products since the last several years. And it's a combination of industry and academics and hospital-based people, and it really works when you put industry together. And one of our focuses is to understand what's going on with troponin, especially high sensitivity troponin. So I took this cartoon off of the HyTest website, and it really kind of sums up their progression and the development and quality production since 1994, 25 years, in the MI prognosis, in the MI diagnosis, and in heart failure. And we'll hear later on after my talk about heart failure. So this cartoon shows that no one really knows exactly, I'll show you a slide, what the exact mechanism per individual, besides when you have a clot in your heart and you get ischemia and you release things. But we learn the more cells that are damaged, the greater the amount of troponin that's leaked from the heart into the circulation. And this could also be a picture of what happens in aging in adults. Because some people think with our new technologies, putting into high sensitivity assays, that we're going to start using age-dependent cutoffs one day in the 60s, 70s, and 80s like we see for natriuretic peptides. So this is the first paper, and the one thing about HyTest, which is really nice, they have a group of scientists that not only do they make things quality products and get these into the marketplace to companies, but they also do real science. And this shows you, we find the date on here. This is 1998. One of the early papers published by the scientific group, and what they were showing here is a lot of different antibodies they developed detects multiple different forms of cardiac troponin I in the tissue. When you choose the best antibody that's going to have the most cross-reactivity, they show that up to 7 different peptides were detected. These are N-C-terminal degradations, oxidative phosphorylation, different forms of I from a 3 subunit to a single subunit complex. And you can see here, you could actually see a serial kinetic pattern that is released. Move forward, 2018, same director, and now new scientists and old scientists, and now with improvements of their technologies, you're now seeing the ability, still the same major form of troponin I, which is the IC complex, is the predominant form. But now they have technologies to look at up to 12 different forms. And now, where the past slide I showed you was looking at changes in the myocardium, it took almost 25 years to get the technology and the ability to detect things in the serum in 5 representative MI patients extracted from their serum. So we've moved with you can imagine the sensitivity with the antibodies, looking at tissue, which has got a lot of protein troponin I. Now we're looking at it in the blood. So I'm not going to talk about mechanisms, but one of the controversies, and my colleague, I see Alan Wu in the back there, we have a little bit of a debate. He thinks there's reversible injury, and I was raised in believing on the literature that the only time troponin is released from the tissue into the blood is cell death. But the mechanisms there are many mechanisms out there, necrosis, apoptosis, cell wound, necropractosis. It doesn't matter what the injury is, but we have the tools now to measure. We don't really know how many cells have to be injured before we can get a signal. That is still an active investigation. Now, a very recent paper that came out of HyTest was just published in Clinical Chemistry, and I'll show a couple slides here because I think this is a great study that will open doors for people to explore. It revisits the concept of what's the latest evidence of what's released and how can we utilize antibodies to look at different forms of troponin, because the goal to put this into clinical patient care is we want to be able to measure any fragment of troponin I to give us a signal that's abnormal. So what they described in this paper is here's the ITC ternary complex. When there's injury, there's C and N-terminal degradation to a lower molecular form. There's further degradation, then you have fragments. And what HyTest has been very successful in doing in the technology they use to develop antibodies, and other people are doing this also, you now can look at the different parts of the protein in the different form, whether it's a full ternary or a binary complex, and they're developing antibodies in different regions of the molecule, again, to try to find out how you can maximize the detection of the troponin when it circulates. Again, another cartoon taken from the HyTest catalog. And this is not a full representation of all the assays, but it's many of the assays in the marketplace or what were in the marketplace. And you get somewhat of a conception here that multiple companies are using antibodies based in this region in the 20 to 40 to 50 range of epitopes. It seems to be a stable region, and you get a lot for your dollar, so to speak. Then other people take different approaches. There might be a sandwich of antibody capture, antibody detection with a signal, or some people now you can see some companies are using 3 antibodies, maybe 2 to capture, 1 to detect, 1 to capture, 2 to detect. And one of the things I thought was really clever that HyTest put forth, and I'll just simplistically put at this, is that remember I talk about the majority of troponin I circulates as an IC complex. So they've shown some work that you maybe could increase your yield of capture of troponin I if you build in an anti-C antibody along with an anti-cardiac troponin I antibody. Cardiac troponin T is not cardiac specific. It's found in both heart and skeletal muscle. But if you're using an anti-cardiac troponin I heart form antibody and you tag it along with in your schematic have a company puts together, you may be able to increase your yield of capture, because sometimes the way the protein folds, you sometimes can't get at certain regions. So I thought it was a very clever idea. The other thing that I actually personally have been involved with some groups in the animal pharmaceutical companies is when you go to do drug development and you want to check for safety, there are animal models initially used before they put a drug into human testing, phase 1. And HyTest has gotten into very carefully looking at different laboratory animals and looking at the heterogeneity and homogeneity of what troponin I and maybe troponin T, and they've developed different antibodies that have different cross-reactivities shown by Western blot. So I'll tell an interesting story. I once did some safety work for a company, and they said in the first phase of the development of their drug, they showed no signal of troponin I, all right? And they were using an assay called DPC. Everyone remember DPC? It's now part of Siemens, all right? When it went to human trials, it turns out the human trials with this drug, the laboratory doing the analysis started using an Abbott assay. Well, the Abbott assay had cross-reactivity with that animal model. The DPC didn't detect that animal model. So all of a sudden they were seeing signals. So it's important that you know what you're measuring, otherwise you can imagine that drug didn't make it because of all the cardiac toxicity that was going on. So this is another really nice field that they've looked at. So this slide I've shown in other talks, we've come a long way. We now have many assays that are cleared throughout the world, and now more and more are starting to get approved in the United States. And what's the impact of these assays? First, the new technologies with the antibody production, they can look at where these, as I showed you earlier, and they can get around any interferences if you pick the right antibodies. So maybe you avoid heterophile antibodies. Maybe you avoid where there's some kind of hemolysis or autoantibodies. So I think the technology can be essentially modeled to allow you to do a crafted way to look at capture and detection antibodies to avoid these known interferences. The other thing that has been described by now 7 different authors in neuromuscular disease, there is some concern that there may be for troponin T cross-reactivity with the cardiac troponin T current assay in the marketplace. HyTest is actually working on anti-cardiac troponin T antibodies. I'm not sure exactly when the IP goes off for Roche, but the early work appears to show that their antibodies essentially don't pick up these same cross-reactivities as shown as this, in this Austrian study that showed false positives for cardiac T without any cardiac involvement where the troponin I was normal. As far as interferences, I have to put a plug in for my committee. We have a table that shows all the interferences as put out by the manufacturers, whether it's hemolysis, whether it's hemoglobin, well, it's skeletal muscle, as well as a list of all of the antibodies used in all of the assays. And we're going to start, I have a postdoc that's going to start not only looking at all these assays for high sensitivity, but we're going to try to figure out which manufacturers and what epitopes are used for every company. And we're going to put a label on, this one's by HyTest, this one's by Siemens, and we're going to try to figure out which antibodies are in each assay. So what are the impacts of all these, assays? Improve analytics, improve lab practices, better for patient care, earlier detection of myocardial injury, that's a good thing. It's not something to worry about. You're going to pick up more injury because it's a bad sign if you have an increased troponin. Early rule out, early rule in within 2 to 3 hours, depending on your algorithm. You don't have to wait 6 to 12 to 24 hours anymore. And the risk is very impressive where we detect things even within the reference range. So I'm not going to spend time on the universal definition. I've kind of alluded to that. But what we're doing now is we're now starting to pick up examples of myocardial injury, which would have been noise before. And this raises flags because if you look at the risk stratification studies, all of these entities portend some long-term risk. So I'll show you, let me show one slide here. And this is going to be the future. So I'm a believer that one day when you bring your children to the doctor before they go to camp for the summer, instead of just getting a cholesterol, I think they're going to baseline your child with the high sensitivity assay. And why? Because if you look at this early study we did in about a 2,000 patient data set, there's incredible risk just looking within the reference range. You can see here, if I can put my glasses on, all right, you can see here that there's a 4-fold difference in risk depending on where your value is, and here's for men. Fortunately, as a 66-year-old, I'm okay still based on the Abbott assay. I'm not have to worry about my risk. We define assays by the ability to high sensitivity. It's an analytic scorecard we've put into place and recommended not only by the IFCC and the AACC Academy, but also the universal definition recognize this, that using the LOD by the manufacturer, what % of men and women have to be measurable, but at least 50% of a normal. Implementing these tests will be, I think, can be challenging, but it's not to be afraid of. It's not a mess. The field is full of good literature. You have to know your assay. But I point out one of the concerns is the troponin T assay by Roche. It's a good assay, but when you get rid of their Gen 4 assay, which is not a great assay, there is a huge increase in the detection of how many increases. While with troponin I assays, most of the contemporary assays are pretty good assays. Switching to T, actually the detection rate goes down because you avoid the noise. So I talked about this at my talk this morning. We've come up with these guidelines that you should look at this paper. It helps you when you implement assays to how to look about both analytics and how it'll impact patient care. As far as point of care assays, we're still waiting for that real, maybe small pocket size analyzer that'll have, or a little small benchtop analyzer that'll have a high sensitivity assay. The antibodies are out there, and I think hopefully we'll see in the next year or two a point of care assay that will be a true high sensitivity. So my final two slides, what I have learned from HyTest in my time before I joined as a board of director, as well as my current here position. The history of their manufacturing of quality and antibodies is as good as any manufacturer out there. It's based on good analytical clinical studies of the antigen that's found in patients' blood, and therefore they have an unmet need to move forward in what they do. As their standard materials were used by the NIST, the National Institute of Standards and Technology, as the standard reference material 2129 when there was AACC groups to help harmonize and get traceability for all assays. They're now introducing recombinant antibodies in different animal models to improve the affinity of their antibodies to, again, eliminate noise and to pick up more signal. And I think you're going to see also when other companies start maybe looking at troponin T assays, they're going to have a quality product, hopefully without any skeletal muscle immunoreactivity. And in our next speaker, we're going to hear really the state of the art of where the company has taken as far as the natriuretic peptides. So I leave you with two last slides, I think. This is a picture of Moscow, Russia. This is the research team headed by the only guy without a white lab coat on. They let you in the room, Alex? Where are you? Okay. He's got a blue sport coat on today, so he looks much better. And finally, this is a picture. I was fortunate enough to study troponin when in January and February. This is a picture from the town of La Scala in Costa Brava in Spain. So this is where I'd like to sit at night and think about cardiac troponin. Not really, but at least I got to think once in a while. So I leave time for about 5 minutes of questions if there's any questions. Thank you very much for listening to me.
Hi, I'm Kristin Eikre from Bergen, Norway. I wonder, what do you think would be the clinical impact of measuring troponin fragments, different fragments? What can it be used for?
So her question had to do with the impact of fragments. Two potential things. One, any troponin released at fragments, we want to be able to measure the total troponin in the circulation. So it increases the signal intensity. So easier to maybe get a signal on a small infarct that has fragments. Make sense? The second part is Jennifer Van Eyck, who's a chemist who's now in the field, she started to say, boy, if we can look at a sequence of timing of the kinetics of troponin, maybe I can backtrack the time to figure out when the actual event occurred. So if I see one fragment now, boy, that's within an hour of an infarct. If I see 6 fragments, maybe that's 3 hours. So the thought was we could do timing over that. And that would be a powerful thing. If you could come in the emergency room, I could get a 20-minute assay that showed me there was 9 fragments in the pattern. The doctor could say, I know how to treat now based on when the infarction occurred. Just a thought.
Hi, Chris Maya from MBio. Actually, threading on that, that suggests that there's actually a role for a multiplex test, something that could look at a couple of different fragments or so forth. What do you see as a desired kind of multi-marker type, collection of troponin pieces, or what's the role of multiplexing in that?
I love the idea. We've actually tossed that around for years. If you could come up with a series of assays that could recognize different fragments and put them onto a microtiter plate, and you could actually get a pattern of signals that define what you'd expect. Remember I showed the slide of 1 to 11 fragments, and you saw you could do some timing. I think that would be a very powerful tool. The key would be to prove that it's going to work. So different antibodies might get different signals, but I think it's a great PhD thesis, to be honest. If you have a multiplex instrument you want to work together, we can talk. Thank you. Good question.